Well, good afternoon, everybody. It's my pleasure to introduce you Steven Bressler. Steven, well, we met for the first time last year in the Society for Neuroscience meeting in San Diego, but at that time we had already 2 papers together. So we knew each other, but
that will be good.
That was very good. Not very good. Well, Steve is professor of the complex system and brain science, and in the psychology at the Florida Atlantic University in the United States. He's interested in cognitive neurodynamics. He's director of the cognitive neurodynamic laboratory there for the Center of Complex Systems and Brain Science. He received a bachelor's degree in Johns Hopkins University, and he got a PhD from Berkeley in California under the supervision of Walter Freeman. And he did a postdoc training in the University of California in San Francisco. He has made a substantial contribution to the field of large-scale neural networks in the brain, with papers cited over 10,000 times according to Google Scholar, and he has published in journals like Nature, Science, and Proceedings of the National Academy of Sciences. He's an active editor for neural networks, and is in the editorial board also in the Computational Intelligence and Neuroscience. He has organized many conferences, including several symposia, etc., in the Society for Neuroscience meeting, which is the most prestigious meeting in neuroscience, in topics including top-down mechanism of visual attention, neurocognitive networks, conference in neural networks, and so on. So he's very active in dynamics. He's does very nice things that we share now. So thank you very much, Steve, for being here.
Thank you for the introduction. Thank you all for coming, and it's a pleasure to be here. I'm going to talk to you about networks today, and these are, for this audience, I think it should be described as complex dynamic networks. So what they also are called is neurocognitive networks. So these are networks in the brain that are responsible for cognition. And so this is probably the main difference that as part of the psychology department and focused on brain sciences, we're interested in cognitive aspects of network dynamics. So maybe that's one difference. I come from a center for complex systems. We focus on brain sciences in particular. So it's smaller than your institute, but we have a PhD program. If you have any students who might be interested in applying, we welcome you to send them our way. Okay, so my laboratory is part of the Center for Complex Systems, And we analyze brain signals from different systems. Over the years, we've analyzed from data from rabbits, monkeys, rats, and humans. And most recently, we're involved in looking at network structure using human functional magnetic resonance imaging. And so this is under the rubric of connectomics, or functional connectivity. And we look at functional connectivity from the point of view of networks and interdependency between brain areas. So that's probably the common thread to all this work, is looking at interdependency between different regions of the brain. And so when we have significant interdependency, then can consider that an edge of a network. And typically in the field of neuroimaging, people are mostly concerned with the activation of individual nodes of the network, or maybe what they call a network would be a distribution of coactive nodes. But we're actually interested in looking at the relationship between the nodes, and this means computing actual quantitative measures for the edges. And so, in my view, you have to describe a network, we need both the description of the nodes and the edges. So it's not enough just to look for a coactivation of a set of nodes. That in itself is not a network. We've looked at field potentials in animals, and EEG, MEG, and fMRI in humans. So we could, I mean, I probably told you everything you need to know already, so anybody who wants to leave, free to go, but if you want to stay, I'll tell you some more about this. Complex dynamic brain networks are large-scale networks. What that means is we're looking at whole brain activity, looking at distributions of activity that are interdependent across different brain regions. So large scale means regional interdependency. So these complex dynamic brain networks are large scale networks. They consist of interacting brain areas, and as I said, the function is thought to underlie cognition. So as a cognitive neuroscientist, I'm interested in the network structure underlying cognition. And by that, we mean specific cognitive functions like attention, working memory, perception, language, decision-making, those kinds of things. So I'm going to tell you about analysis of functional connectivity, and primarily directed functional connectivity, or DFC. And we'll use 3 examples, 2 from human, one in human visual spatial attention, the other in human motor coordination. And then the third line of evidence comes from rhesus macaque monkeys performing a visual pattern discrimination task. So the idea is that these networks are coordinated in preparation for execution of a task, as well as being coordinated during the task. So one of my main take-home messages will be that we have to be concerned about these preparatory processes, as well as the processes going on during the execution of the task. And the idea for this approach came from a Russian neuropsychologist, Luria, And in his book in 1962, he said that the concept of localization of functions has come to mean a network of complex dynamic structures or combination centers consisting of mosaics of distant points of the nervous system united in a common task. So this is a very different definition of localization of function than was traditionally thought of. And here localization means a network, and this is a complex dynamic network. It's changing. It's complex because there's many interacting parts, and these parts are distributed across the whole brain, both cortical and subcortical. So I've written extensively about these networks, and I welcome you to look at some of the references. The idea here is pretty much the same. So part of the dynamics of these networks is that they are functionally organized by the processing demands put upon the brain by the particular cognitive function. So when you're engaged in a cognitive task, there's a certain load imposed by that task on the brain. And so the functional organization of the network is a response to those processing demands. And I'm going to argue that the functional organization is further constructed by mutual constraint imposed on the constituent neural populations that are interacting as part of the network. And let's see, I already covered edges and nodes. So functional connectivity has been studied in brain data, and it's usually applied as an undirected measure. So correlation in the time domain and coherence measures in the frequency domain. But as I said, I'm going to tell you about analysis of connectivity that's directed. And to measure directed functional connectivity, we need time series. So we, even with the fMRI data, we treat the signal coming from every voxel as a time series. And there are methods that have been developed for measurement of directed functional connectivity from time series. And this can be either in the time domain or the frequency domain. So the main measure that we've used is Wiener-Granger causality. And this can be measured by using autoregressive models applied to time series of the neural data. And I'll tell more about that as I go along. In the frequency domain, we measure this in local field potentials and EEG. And in the time domain, we measure this in the functional magnetic resonance data. Okay, so the idea for directed functional connectivity can be traced back to Norbert Wiener, who in the 1950s said that for two simultaneous time series, one series is called causal to another if we can better predict the second series by incorporating knowledge of the first. So that was the idea that came from Wiener, and it had to do with predictability. And so even now, all the subsequent developments, the same basic idea applies. So people often get confused with this. The idea of causality in the Wiener-Granger sense is not the same as a direct physical causality. So by causality, we just mean what Wiener said, that it's based on time series predictability. And better prediction means causality. And that's all there is to causality. Well, Wiener proposed this idea, but he didn't have any implementation in mind. Or he did, but it was in terms of this very kind of esoteric and abstract mathematics. Granger came along in the next decade and formulated the idea of Wiener in terms of autoregressive models. And it became very clear what the formulation would be. So we've taken this idea from Wiener and Granger and applied it, and the first line of evidence that I'll tell you about is work with fMRI in what's called the dorsal attention network. So people knew about this network before and postulated these interactions between the different parts of the network, but it had never been measured before. So the actual arrows that were drawn here are fictitious. They are just suppositions about what is interacting with what's causing what. And so nobody had actually measured. So what you could say probably is that there was an idea of what the main players were, but nothing was known about the interactions before about 10 years ago. So what we found when we started to apply the directed functional connectivity analysis to this data was that this dorsal attention network exerts a top-down control of another network in the visual cortex, visual occipital cortex, in visual spatial attention. And so what is called in psychology top-down attention is actually implemented in the brain by top-down influences coming from this dorsal attention network and being applied to various parts of the visual occipital cortex, specifically in visual spatial attention. So it was known that the areas of the dorsal attention network, which consist of frontal and parietal regions, were consistently activated by cues. And this was as part of a Posner spatial cueing task. We'll have more about that. But it wasn't known what the edges of the network were really. There was just speculation about what the edges might be. So in this experiment, the subject receives a preparatory auditory cue, and then there's a delay period in which the subject is directed either to the left or to the right upper visual field. And so they're attending to one of these fields, expecting or anticipating that a stimulus will appear in the field and another stimulus will appear in the other field, and their attention is directed just to the cued location. So there's a report cue that says that either they should keep their attention on the cued location or switch it. That's part of the Posner task. But in our case, we just were interested in trials where the attention was at the cued location, and the task was to be applied there. So there was a report cue that said either left or right. It was also auditory, and there was a delay period, and in which the subject is attending to one of these locations. But this stimulus with the grating doesn't appear until some 10 seconds later. And the bold signal is recorded in this intervening time period. So we analyzed the directed functional connectivity in this period here, where the subject is cued but has not received the stimulus for processing. And what we found is that there was a consistent top-down influence by way of directed functional connectivity, and it was found for all of the subjects in the experiment, and it was found for all except one of the pairs of ROIs, one pair being in the dorsal attention network, the other one voxel location being in the dorsal attention network, the other being in the visual occipital cortex. And this went away when we used a trial randomized control or a voxel randomized control. Either one, this disappeared. So there was a distinct asymmetry that we found with the directed functional connectivity analysis, showing a top-down influence from the dorsal attention network to the visual occipital cortex, which makes sense. When you're doing this task, you're cued as to the location, and then you're just waiting for the stimulus to appear so that you can process it. And it had been speculated that there was a top-down influence exerted by the frontal cortex on the sensory areas in anticipation of processing the stimulus, but it had never been shown before. So this was the first direct evidence for that. More recently, we've followed up with this same data and again found that top-down influences from the DAN to the VOC are consistently greater than bottom-up influences. That's what we saw before, but now we could evaluate this across the time of the trial. And with recent advances in the analysis, we show that this persists after this top-down greater than bottom-up, this asymmetry in the directional functional connectivity persists after the onset of the target. And so that suggested that what we're seeing is a preparatory set which is induced in the subjects as a result of their cueing for performing the task. So the top-down is consistently greater than the bottom-up for both DAN to VOC, and also from the part of the frontal cortex, which is part of the dorsal attention network, to the parietal part of the network. There's also this top-down greater than bottom-up asymmetry. In more recent work, we've looked at similar relation between dorsal anterior cingulate cortex and supplementary motor area, DACC and SMA, and found that when subjects are doing a motor task, a visual motor task, that there's also an asymmetry in the relationship. There's a top-down greater than bottom-up asymmetry. That is, the influence from DACC to SMA is greater than in the reverse direction. We also, most recently, in a paper that was just published, found that there's a reversal of this asymmetry during the rest period. And in a typical fMRI task, there are blocks of task separated by blocks of rest. So we compared the rest period in this visual motor coordination task to a rest period in a control, a working memory task, and found that the rest periods were different in that the bottom-up influence, that is from SMA to DAC at rest, was consistently greater during the coordination task blocks than during the working memory control task. So this suggested that not only is there a top-down influence, but it might be potentiated during the rest period by a reverse directed influence. And as I said, this just came out this year, so we're still working on this. But this is potentially interesting that the bottom-up influence can be greater during rest. Okay, that covers the human work. And now I'm going to turn to the monkey field potential work. And here, monkeys were doing a visual pattern discrimination task. And a typical Go trial is represented here. And the analysis was conducted in a pre-stimulus time when the LFPs that were recorded from multiple sites in one hemisphere were relatively stationary. And following the stimulus, there's this non-stationarity which has to be removed before the analysis can proceed. And that's because the autoregressive models that this is based on require stationary stochastic processes. So we applied the analysis just to this pre-stimulus part. And the implementation of the Wiener-Granger causality could be made in just a short time window before the stimulus came on. And that's in a time period when the monkey has pressed the lever indicating a engagement in the task, but the visual stimulus has not appeared. The task was simply to discriminate between line shapes and diamond shapes. But in this period that we analyzed, the monkey is just waiting, just like in the humans were doing in the visual attention task. Okay, so in the first thing that we studied was the somatosensory motor system. That's been published, and we found networks of coherence using undirected functional connectivity and networks of Wiener-Granger causality using directed functional connectivity. And this work was different from the human work in that it was carried out in the frequency domain, because the one advantage that the monkey field potential provides is the greater temporal resolution. The human fMRI recordings are made only with a time step of like 2 seconds. And that's an eternity when it comes to doing these cognitive tasks. And so the resolution here is on the order of 1 millisecond, much finer temporal resolution. And so we could make autoregressive models that were longer in time, and we could do frequency analysis on them. And so this, what we observed was spectra of Granger causality rather than single scalar values as in the human fMRI analysis. So in the somatosensory motor system, there's a pattern of undirected or coherence between sites, and also directed Wiener-Granger causal influences between sites. And so that reflected the loop that the monkey was going through in pressing down the lever and waiting to process the task. Here, we're looking at the visual cortex and looking at pairs of electrodes that are synchronized in the beta frequency range. So this whole frequency analysis that's possible in the monkey allowed us to localize in the frequency regime where the rhythms were prominent. And it turned out in the coherence and in the Granger causal network, there's a prominent rhythm at low beta frequency, around 18 cycles per second. And so here's shows all of the electrode pairs from 2 monkeys, and there's a prominent peak in the Granger causality spectrum, but only in the top-down direction. That is from V4 and TEO to V1. In the bottom-up direction, there's no peak. So the arrows here represent the strength of directed connectivity, and they're all pointing in the top-down direction, reflecting this peak in Granger causality. Both the somatosensory motor system and the visual system, as well as the prefrontal cortex, which we've also analyzed, show this beta rhythmicity. And so in this task, there is beta synchronization across the multiple systems in the cortex of the monkey. In the motor system, the beta frequency is a little bit higher. It's in the 20 to 25 hertz range. In the visual system and the prefrontal system, it's lower. It's in this 15 to 20 hertz range. We did a pattern discrimination because there were different rules in this task, and the monkey had to associate either the line stimulus with the go or the diamond stimulus with the go. And the rule for correct behavior would change from session to session, and the monkey had to learn the rule that was in force. So we had trials which were identical except for the rule, and the rule determined the correct response for a particular stimulus type. And so we could do pattern classification, and we found that there was about 80% classification in this red distribution. We used a support vector machine to do the classification, and we used the pattern of pre-stimulus top-down beta band Granger causality as the features for classification. The blue distribution just shows a bootstrap distribution of what's expected by chance with randomization of the labels. So this was a demonstration that this top-down beta band Granger causality was carrying the context of the task. And keep in mind that all of the Granger causality was measured within the visual cortex. So it was either extrastriate cortex or V1. And yet the behavior of the task was reflected in this top-down influence. The whole pattern of top-down influence was involved and allowed us to do the classification and get significant classification. This distribution has to do with the bootstrap procedure that was followed. So, I'm doing pretty good for time. Take-home message is that task execution for a variety of different tasks, both in humans and monkeys, depends on the functional organization of these complex dynamic brain networks. And they do appear to be organized according to the cognitive task that's being performed. The coordination or synchronization that we see between the nodes of the networks occurs not only during the task execution performance, but it also occurs in preparation for task performance. And if I had to bet, I would say that this preparatory coordination is what makes the human behavior different from machine behavior. And if we want to make artificial neural network models which are more versatile and powerful, we will have to include this preparatory coordination, which we've seen in the both the human and the monkey data by way of this connectivity analysis. So it's the preparation which distinguishes human behavior as it currently is from machine behavior, and incorporating preparatory processes by way of coordination of different parts, I think will make artificial neural network models more powerful. Thank you.
Thank you, Steve. Ingo.
May I pick up on your last point, the last take-home message?
Sure.
You say that the artificial neural networks might profit from preparatory coordination. That would require to have expectation, right? So that the network would need to expect a certain signal. How would the artificial network do that? So it doesn't the cat bite its own tail.
Yeah. Well, expectation comes from memory. And so we have to use prior knowledge that's stored in memory to devise expectation processes. But if you think about it, what your own behavior is, at least half of human behavior is expectation. You prepare to do something as well as do it. And a lot of cognitive neuroscience deals with this preparatory processing. And so what the prefrontal cortex in particular does is look into the future and prepare for different courses of action behavior. So we need, I don't know how you implement that, but that's what is needed. And I think that once machines have that kind of preparatory ability, then they will be more like humans in being able to produce flexible and more powerful behavior.
You have talked about networks, but I've seen most of the results are of pairs of nodes. So you compare one voxel in one place with another voxel in one place, and you understand what is the directionality of the position.
Yeah.
Just how do you determine which voxel to choose? And there is no.
Well, in that.
There is no feedback. Perhaps there is another voxel which is connected in the other direction. So it's how do you determine the voxels, and it's so clean, the top down, and nothing of in the other direction?
Yeah, it appears that, well, we know that there is feedback during processing of tasks, and it appears that in preparation there is a stronger unidirectional bias. I don't know why, but I would expect something in the bottom up, and there's nothing at all. It's just weaker than the top down. So you can have feedback that's not of the same strength as the feed forward. That's one thing. Another thing is that you asked about the voxels. So we construct a network by looking pairwise, that's true, but we look at all possible pairwise combinations. I should have made that clear. If you have a set of nodes, and in the first experiment and in the second experiment, we were focused on, and in the third experiment, in all of these, we're focused on a particular set of nodes. So we have a set of areas that are interacting to start with, and then we look at all pairwise combinations. So it is pairwise, but we look at all the pairs. And not only do we look at all the pairs, but we look at both directions for every pair. So you can compute the Granger causality from X to Y and also from Y to X. And we do that so you can construct a network pairwise. Maybe there's other techniques that go beyond pairs, I don't know. But you can construct a network.
My point was on the selection of the...
We don't just...
Now that there is some part of the brain that your hypothesis that this is the one controlling, and you have the hypothesis that this one is responding, and then you compare
no,
those
there's
things.
no...
Don't you compare the flux of information inside, let's say, inside one of those networks?
No, this is all large scale, so it's just looking at these distributed nodes. And we look at all the pairwise combinations. So for the functional connectivity analysis of the human fMRI data, we had to go from voxel interactions to ROI interactions. And so there's a way that we have, and that's been documented, we have a way of doing that. So then, but even there, we look at both directions between all pairwise combinations of ROIs. So I guess the answer to your question is that we can form a network pairwise, and we would expect there to be feedback. And in the text execution, I think there is feedback. But for whatever reason, it doesn't seem to be in the preparatory process. It seems to be dominant in one direction.
I have a question. This top-down interaction that you find is done after the monkey is trained. What would you expect to happen during the training process? Would that exist already before they are trained to do this task? Or?
Well, it depends on the task. Some of these behaviors are very naturalistic, and for those kinds of behaviors, then I think there's a circuitry that is already trained in the monkey. But for higher order tasks that are more novel, then I would expect a naive cortex initially, and that it becomes tuned after training. And there's some evidence already from functional magnetic resonance imaging that the top-down influence does change as a result of learning. And so with learning a new task, the implication is that this naive cortex will become tuned or trained and will expect input from other regions. So each node will have an expectation of what it's going to receive from other parts of the network, and that expectation is something that's learned. This has to do with the memory of the system, what the system has experienced. So as the monkey is experiencing new inputs and making connections to responses, the cortex is also becoming rewired, so to speak, to support that new behavior.
Question? Yeah.
Is a repetition equivalent to preparation? I mean, if you do the task the second time, are you ready as much as you was prepared from an external stimulus?
Well, there is something called memory solidity, and repetition is one of the factors that contributes to the solidity. And so I would expect, this is just inference, but I would expect that the expectation process, because of this memory solidity, will become more solid, so it would be stronger with further repetition of the trial. And as learning progresses, then the expectation should develop and become stronger. So I haven't ever seen anybody talk about solidity of expectation, but one could infer that there would be a solidity of expectation that paralleled the solidity of memory. Memory is a big factor in contributing to the expectation. That's a good point.
Any other question or comment? But if not, thanks him again. It will be
Tried to speed through it. I guess I went a little bit too fast. Take up the whole hour. Okay.
Sure.